Howdy, technical science. We're going to look at moving in and out of cells.
I could while away the hours, conferring with the flowers, consulting with the rain. And my head I'd be scratching while my thoughts were busy hatching, if I only had a membrane.
Thanks to Sam Moore for producing this PowerPoint. So science understanding, we're going to look at substances move in and out of cells by diffusion, osmosis, active transport, endo and exocytosis, and explain how the structure of the membrane allows this to happen, and talk about how movement is affected by surface area to volume ratio, concentration gradients, and the nature of the substances being exchanged. So let's talk about the structure of the cell membrane. So it's a phospholipid bilayer with protein molecules embedded in it, and we call this the fluid mosaic model. So there's two layers of phospholipids. So here's the top layer, here's the bottom layer, and then embedded in that is a whole heap of proteins and other structures as well. The proteins might be involved in transport, they might be involved in communication as well. Here we have a carbohydrate, for example, that might be involved in communication. One end of the phospholipid is hydrophilic, that means it likes water, so that points towards both the intracellular and extracellular environment. Then we have a long carbon tail, which is hydrophilic, which doesn't like water, so that points in towards the other carbon tails from either end. So we have hydrophilic on one end, hydrophobic on the other. They will produce this structure without energy. So what does the cell membrane do? So it separates the inside from the outside of the cell. It can allow substances in and out of the cell. It can also allow for recognition, and it can also allow for the cytoskeleton to attach to it. There's also receptors there that allow for communication. So that was that polysaccharide that we saw before, the carbohydrate. So the cell membrane can allow there to be different concentrations of substances inside and outside the cell, and this can be important for driving particular chemical reactions. So if we have a look down here, we've got some ions, for example. We can see inside the cell there's a great difference in concentration of sodium compared to outside. So outside is a lot higher than inside. What this means is that the cell will have to constantly pump these sodium ions out to maintain that difference in concentration. If we look at potassium, it's the other way around. So there's a higher concentration of potassium inside than out. So in this case, they have to pump potassium inside the cell and then pump sodium out. So there's a sodium-potassium pump that does this, for example. So a pump is a protein structure. We're going to look at that in a second. If these concentrations get out of whack, then the cell can get into a lot of trouble. So let's talk about some of the ways that substances can move. So one of them is diffusion. And diffusion is where a substance moves from a region of high concentration to a region of low concentration. We say this is moving with the concentration gradient. So if we can think of one side as being high, one side as being low, you're moving with that gradient from high to low. If we have a look at our sheep has done a fart. That fart, we have a high concentration of poo gas in one place. As time goes on, we move from high concentration to low concentration, so the air around it. So this is a good example of diffusion. Now, diffusion continues until equilibrium is reached, so the same concentration in your space. It doesn't mean that the movement of the particles stops, it just means that the concentration doesn't change from one place to another. So once we've reached equilibrium, there's the same concentration everywhere, there's no concentration gradient. So with our sheep fart, so the sheep farts, the molecules move out, and then they keep on moving, but we get the same concentration over the same amount of space. Diffusion is passive, it doesn't require energy from the cell. So passive is not energy from the cell. It does require energy from the environment, but it doesn't require energy from the cell specifically. It's important to note that with these transport processes, it depends on what substances you're moving as to how well they'll move with the process. So small molecules will move a lot easier than large molecules. Molecules that are charged will have a lot more difficulty than molecules that aren't charged. And if something is a lipid, it will make its way through the membrane really easily because the membrane itself is mostly lipid. So diffusion occurs in a cell through the bilayer, and it's not affected by the concentration of other substances other than the substance that you're looking at. So we have to run with a concentration gradient. So osmosis is diffusion of water. So it's the movement of a solvent from a region of high solvent concentration to low solvent concentration. But that doesn't really work because solvent concentration doesn't make sense in terms of chemistry. So osmosis is the movement of a solvent, like water, from a region of low solute concentration to a region of high solute concentration. I think that's a better way of thinking of it. So osmosis, too, is passive. It doesn't require energy from the cell, it just requires energy from the environment. So one way of looking at it is that it's moving from a watery solution to a less watery solution, or you can look at it in terms of the solute concentration. So from low solute concentration to high solute concentration. This can generate what's called osmotic pressure, and it can help maintain the cell shape. You need to have a certain amount of water inside the cell. And for plants as well, you need to have a certain amount of water inside the vacuole to maintain the shape. If the osmotic balance isn't right, so if the concentration of solutes isn't correct, so in an animal cell, if too much water goes in, the cell will burst. And in either cell, if there's not enough water, the cell will shrink, and that might be killing it. If a cell is full of water, we call that turgid, and this happens with plant cells. Cells that don't have enough water we call flaccid. So here's some terminology: hypertonic, isotonic, and hypertonic. So in an isotonic solution, there's the same concentration of substance on each side of the membrane, or the semi-permeable barrier. So here we have water going in and water going out, but it's going in and out at the same rate, so the cell doesn't change size. So a hypotonic solution has a low concentration of solute compared to the inside of the cell, because we're talking about the solution outside. So there's less, for example, salt outside than inside. Water will go where there's more salt, or where there's more sugar, or where there's more of anything compared to the outside. So in this case, for a blood cell, the water will come in, and that will cause the blood cell to burst, and we call that lysing. In a plant cell, this is called being turgid, so water comes in and that puffs out the cell wall, since there's more water inside. In a hypertonic solution, you have a higher concentration of your substance outside the cell compared to inside, or outside your semipermeable membrane compared to inside. So in this case, it would be saltier outside. Water goes where it's saltier or sugarier, higher concentration of your substance compared to the inside. So in this case, water comes from the inside of the cell out. This shrivels up the cell, and in a plant cell we call this plasmolysis. You can see the cell membrane pulls back from the cell wall. So repeating a key point from before, not all molecules are able to diffuse through the membrane equally. There's charge, there's size, and there's solubility as well that affects this. To help substances move, we can embed proteins in the membrane that allow some substances to come through faster than they normally would. So they might act as gates or carriers to allow substances to come through the cell membrane that normally wouldn't be able to. So this is why the membrane isn't just semi-permeable, which means it allows some stuff through, but it's also selectively permeable because it can allow certain substances in and out, depending on what the substance is, so it can select what comes in and out of the cell. So we can have a process called facilitated diffusion, where we have a protein that allows substances to come through the membrane a bit easier. So substances that might be too big to normally diffuse through the cell membrane, or substances that might have a charge, or that might not be lipid soluble, or are lipid soluble, or too lipid soluble. So here's a channel protein, so it's allowing a large molecule to come through from the outsides of the cell to the inside of the cell. So again, this is a diffusion process, so it doesn't require energy from the cell, so it's passive. So this can allow molecules like glucose, which are quite big, or amino acids, which too can be quite big, to move through, but they're moving with the concentration gradient as well, so they're going from high to low concentration. So the processes we've talked about previously have been passive transport. They don't require energy from the cell. Now we're going to talk about active transport, which does require energy from the cell. So in this case, energy, usually in the form of ATP, is used to change the shape of a protein, and that allows substances in or out of the cell. Now, this is one type. There are several types of transport that are active that require energy from the cell. So this kind of structure here we might call a pump. And it's a pump because it requires energy, and you're going against the concentration gradient in this case. So here we have a low concentration outside and a high concentration inside. We are using energy to increase the concentration inside compared to outside. So normally these molecules will be flowing out in that direction from high concentration to low concentration. But using energy in the form of ATP and these pumping proteins, we can increase the concentration. We can go against the concentration gradient from low to high. So now we'll look at some specific types of active transport that don't involve membrane proteins specifically. So endocytosis is an example. In endocytosis, the cell engulfs particles and takes them into the cell. So the cell membrane is used to engulf the substance, and then that is pulled inside the cell to produce a vacuole. There's two types. There's phagocytosis and pinocytosis. So phagocytosis, so here we can see the cell membrane. It's forming a vesicle around the food particle in this case and drawing it inside. Now because there's going to be a whole heap of movement of the cytoskeleton here, that requires a lot of energy from the cell, so it's an active process. Pinocytosis is taking in substances normally that we think of as dissolved in a liquid. So it might be ions, it might be very small particles, but it's really sucking in things. So it might be sucking in fat droplets from the outside, for example. But again, so the membrane forms a little pocket that sucks in some of the extracellular fluid. The vesicle forms when the membrane pinches off, and then that's taken into the cell. It's just saying that again. So phagocytosis, so where you're taking a large molecule in, a large food molecule, for example. So it can be taken into the vacuole. We can get a lysosome, which contains digestive enzymes, merging with the food vacuole. The digestive enzymes are released in there, and that can break down the food particle. This is how amoeba work, this is how macrophages work, but it's important that this is selective. It's not just any old thing that's going past. The cell decides what it's going to take in through phagocytosis. Pinocytosis, like I said, happens on smaller scales. That's normally, if we think of it as cell drinking, so it's taking in smaller substances, normally liquids, and substances in the liquids that the cell wants. So large droplets, for example, of fat, like I said before, these can be taken in the small intestine, for example, by pinocytosis. So phagocytosis is specific, where particular food particles are being taken in, or particular organisms are being taken in, Endocytosis is a bit more non-selective because you're just sucking in all the extracellular fluid that's just directly outside your cell. Last bit is exocytosis, and this is the reverse process of endocytosis. So rather than taking substances in, it's taking substances out. So exo means out, cyto means cell, so out of the cell. So in this case, vesicles, which are packages of cell membrane basically, they might contain something like a protein or lots of proteins. They merge with the cell membrane and then disgorge their substances to the outside world. So they're balls of membrane that contain substance. They merge with the cell membrane and kick it out to the outside world. The Golgi apparatus is responsible for a lot of this. So we talked about, when we talked about the structure of Golgi bodies, how there's vesicles that are formed. So those vesicles make their way to the cell membrane and then disgorge their products. Cells that make large amounts of substance for export, like salivary cells, for example, making proteins in saliva, and hormone cells, they have large numbers of Golgi bodies, so they can do a lot of this disgorging of substances out to the extracellular environment. So here we can see this. So here we have a Golgi body. We can see that we have this vesicle. It's got substances inside that we want to kick out of the cell. So these might be proteins that might be required by other cells. The vesicle is produced, it makes its way to the cell membrane, and it disgorges its products to the outside. And again, there's going to be a whole heap of movement of the cytoskeleton to produce this, and that requires energy from the cell. So this is an active process. So today on Flippin Science, we looked at transport through the cell membrane. That's the Flippin Science today. See ya.